All right, hi everybody. We're going to be talking about white blood cells in this lecture. As you go through this, it's best to just, as your textbook has, build up a table just describing all the different white blood cells, what their physical characteristics are, how you can visually identify them under a microscope, and obviously, perhaps most importantly, tell what they're actually doing, what their actual function is. So this is a slide that was shown in Biology 1160. So this is showing you basically hematopoiesis. Everybody should remember the term hematopoiesis from Biology 1160. This is the generation of all of the blood in your body. So, and you remember in adults this happened, for example, in the red bone marrow, in the epiphyses of long bones, for example. So we start with the pluripotent stem cell. Pluri means it can just give rise to everything. There are two different daughters of the pluripotent stem cell. One is called the myeloid stem cell, and one is called the lymphoid stem cell. Now there's some additional information here in green that you don't really need to worry about for this course, but I want you to appreciate that the lymphoid stem cells give rise to very specific population of cells. These are the T cells, the B cells, and the natural killer cells. The T cells and the B cells are referred to as lymphocytes, and we'll be talking about them at length in a couple weeks' time in this course. All of the other blood cell types derive from the myeloid stem cell. So this includes your red blood cells, your platelets, your mast cells, which we're really not going to talk about very much in this course, and then the remainder of the white blood cells, the eosinophils, basophils, neutrophils, and monocytes/macrophages. So what we're going to do for the remainder of this lecture is just give a brief overview of each of these white blood cells and set the stage for the descriptions of what's going to happen in the next couple weeks in this course. So let's start with the most abundant of all white blood cells, which are the neutrophils. So 60 to 70% of your white blood cells are neutrophils. If we were to give you a slide like we would have if we weren't in COVID and social distancing, if we were going to the lab, you would sit at a microscope and you would look at a prepared slide that has been stained for white blood cells. And what you would see for the white blood cells is that there would be a lot of neutrophils and very little of everything else. So neutrophils have a distinctive multi-lobed nucleus. So you can see the nucleus kind of not as like a perfect circle as like you're used to seeing. You also notice that there are some granules, these spots that are all over the surface of the cytoplasm of these cells. So we call these granular white blood cells. The reason they're called neutrophils specifically is not of any importance other than when researchers first found them, they found that these cells specifically stained with neutral dyes. So it's more of an artifact of the experimentation process. In terms of functionality, neutrophils carry out phagocytosis, which I've described at length already in this week's work of lectures, and they also have chemical destruction of microbes. The neutrophils are like the first to arrive at the scene of a crime. So if there was a wound on your body that was incurred, damage to blood vessels, a bacterium establishing an infection that's causing damage to the cells around it, these things would attract neutrophils. So the neutrophils are what sort of go to the area where the danger is, and then they phagocytose or destroy whatever they come into contact with. And then once they phagocytose, they can then go back to the immune system and sort of alert the immune system to what the specifics of the threat are. Eosinophils, which are far less abundant, 2 to 4% of all white blood cells, they are also granular, as you can see. All these granules all over the white blood cell here. And sort of conversely, compared to how the neutrophils were stained by neutral dyes, eosinophils are stained by acidic dyes. And you can see kind of this neat bilobed nucleus here. So in terms of functionality, these are sort of antihistamines. So these are what are responsible for combating the effects of histamine. And when you think about histamine, you should think about this term inflammation. And we'll talk about inflammation in detail next week in this course. And I do have another slide that I'll talk about in a second dealing with inflammation. So eosinophils are what tries to turn off your your inflammatory pathway, which is obviously very good for your health, so long as the inflammatory pathway has already been successful in doing what the inflammatory pathway is supposed to do. Although this doesn't influence us much here in Canada, certainly eosinophils actually have a secondary function in terms of attacking parasitic worms that are trying to establish a parasitic relationship in our body. And eosinophils can carry out phagocytosis of antigen-antibody complexes. So this happens a little bit later. So when antibodies have been produced and attached to an antigen in this case is anything that's foreign in your body. But more particular interest to you, probably something that is microbial, some sort of potential pathogen. And if it's already bound to an antibody, then there's already been some measure of an immune response taking place in your body. Basophils represent 0.5 to 1% of all white blood cells, so much less than the other ones. They are granular as well. You can see the granules here. Their nucleus is a little bit harder to see. They are defined by being stained by basic dyes. That's where the name baso comes from. And their functions are to release the inflammatory chemicals like histamine. I'm not going to talk too much about what inflammation is doing on a fundamental physiological level this week, because we're going to talk about it at length next week. I'll just leave you with this. I will describe this next week as well, but there are 4 words in Latin that are used to describe inflammation. Rubor, which is redness. Calor, which is heat. Tumor, which is swelling. And dolor, which is pain or loss of function. And those are the 4 signs and symptoms of inflammation. And as you know, histamine is what's responsible for initiating that inflammatory process. And as I mentioned on the previous slide, eosinophils are involved in turning off or turning down that inflammatory process. Oftentimes, people with bad allergies are going to be compromised at the eosinophil level. Monocytes and macrophages are the first type of agranular leukocyte. They have a distinctive horseshoe-shaped nucleus that you can see here. But you notice they don't have any granules in their cytoplasm. And their function is entirely phagocytosis as a fixed or wandering macrophage, which is what they act like in a mature form, which are these sort of amoeba-like cells that I talk about all the time that are wandering all the different tissues of the body, like little vacuum cleaners, searching for foreign bodies to digest and then ultimately bring back to the immune system to alert our bodies to these foreign invaders being present in our body. Finally, we have the lymphocytes, which are representing, next to neutrophils, the largest proportion of white blood cells in your body, 20 to 25%. They are agranular. They have sort of this mostly round nucleus with a little indent in it. The 2 types of cells that are lymphocytes are called T cells and B cells. We will cover this not next week, but the week after, because they are specifically involved in the adaptive or acquired immune response, which, remember, the name of the game for the adaptive and acquired responses is specificity. So this is the very targeted way in which our immune system attacks an infectious agent in the most specific way possible. It's a very complicated process, and that's why we're going to dedicate a whole week's work towards it in a couple weeks. All right, that's the end of this lecture, and I'll see you soon.